the sun. It provides energy in the form of light, light our Earth needs to sustain itself, to support a system in which plants and animals convert, then use the energy that fuels the world of life. In this ongoing system, plants harvest light and convert it into chemical energy. This energy can be used by the plant or harvested by other organisms. It's a delicately balanced relationship of energy in, energy out. To maintain the properties of life, organisms harvest the light energy produced by the sun. This energy enters the world during photosynthesis, a life-sustaining process carried out primarily by green plants. Using this process, plants produce organic molecules that store the energy. The energy is then available to other organisms in the food they eat. After it's used by organisms, the energy leaves the living world as heat.
Photosynthetic organisms are referred to as photoautotrophs in the sense that they are able to use light energy to form the basic building blocks that they need to synthesize important organic molecules.
What do you have here? I have DCMU.
Dr. Jean Erickson studies a single-celled alga. It too, like the grass in your lawn, photosynthesizes.
I think that's great.
Photosynthesis is arguably one of the most important chemical reactions on Earth. All life as we know it on Earth is sustained by this process in which sunlight is captured by plants and converted into chemical energy. Leaves are the part or organ of the plant that are particularly well adapted for photosynthesis. The upper and lower layers of the leaves are composed of epidermal layers covered with a waxy cuticle that prevents loss of water, but also, of course, prevents infusion of carbon dioxide into the leaf.
Since photosynthesis requires carbon dioxide, the epidermal layer is equipped with specialized structures called stomata. Stomata are pores that allow gases to pass into and out of the leaf. Below the epidermal layer are cells containing chloroplasts, the organelle responsible for the photosynthetic process. Without chloroplasts, plants could not harvest the light energy essential to life on our planet. Within the chloroplast are thylakoids, internal membranes often seen stacked like coins. The light-trapping pigment molecules and their associated proteins are carefully arranged in the thylakoid membranes.
Each chlorophyll molecule is specially designed at a chemical level such that when a photon or light energy hits that chlorophyll molecule, an electron is raised to an excited energy state. And then, because of the arrangement, tightly packed chlorophyll molecules in the leaf light harvesting complex will transfer that excited energy state to a neighboring chlorophyll molecule. So this is the harvesting of light energy. And all of that harvested energy then is funneled to a very special chlorophyll molecule at the heart of what we call the photosynthetic reaction center.
The light-dependent phase of photosynthesis involves two groups of light-absorbing molecules found in the thylakoid membranes. Called photosystem 2 and photosystem 1, they are clusters of light-trapping molecules which funnel energy to special trap molecules. When excited, these special trap pigments release energized electrons. An acceptor molecule picks up the energized electrons and passes them down transporting molecules in the thylakoid membrane. Due to this passage of electrons, hydrogen ions accumulate inside the thylakoid. The energy from this proton gradient is harnessed to synthesize energetic ATP. In an all-important phase, the splitting of water replaces the electrons lost by photosystem II's trap molecule. This step is critical because it generates hydrogen ions for the gradient. It also releases the oxygen humans and other animals need to survive. Responding to light of a different wavelength, photosystem I's trap molecule ejects energized electrons. These electrons are picked up by different carriers until they are added to a molecule of NADP. This forms the second important product of the light phase, NADPH.
Our research is focused on understanding how this pigment protein complex of photosystem II is able to harness the sun's energy to split water into oxygen, electrons, and protons. This is arguably one of the most important chemical reactions on Earth.
For the plant, the oxygen released by this process is a byproduct or waste. What's important to the plant is that the released electrons energized by light make ATP.
In the conversion of light energy to chemical energy, there are two important molecules that store chemical energy. One of these is NADPH, the other is ATP.
While the light-dependent phase makes NADPH and ATP, the dark or light-independent phase uses them both to produce sugars such as glucose.
The so-called dark phase reactions of photosynthesis refer to the fixation of CO2, inorganic carbon dioxide, into organic carbon. And this takes place in the stroma of the chloroplast.
The Calvin cycle, the dark phase of photosynthesis, produces sugar, a valuable fuel for most organisms. This cycle occurs in the stroma or fluid portion of the chloroplast. Here, an enzyme adds carbon dioxide to a 5-carbon compound and forms an unstable molecule, which immediately divides into a pair of 3-carbon compounds. These molecules are energized by reactions with first the ATP and then NADPH produced in the light phase. In the dark phase, some molecules undergo a series of changes, using additional ATP to reform the 5-carbon compound with which the Calvin cycle started. Others combine to form the end products of photosynthesis, the basic fuel for most living organisms, 6-carbon sugars.
Energy flows through photosystems 2 and 1 of the light phase and is converted to 6-carbon sugars in the dark phase. Photosynthetic organisms harvest inorganic carbon dioxide from the atmosphere and convert it into organic carbon, producing simple sugar and releasing oxygen and water. These sugars are not only used by the plant, they provide essential nourishment for plant-eating animals. Sustaining the flow of energy, animals and other organisms burn the sugars, then release carbon dioxide and water, a process that frequently takes place in the mitochondrion, the powerhouse of the cell. All organisms have the ability to release energy stored in sugar and other organic compounds. And in most organisms, aerobic respiration is the main energy-releasing pathway, a process that requires oxygen. Unlike plants, which are autotrophs, animals and other organisms are heterotrophs, meaning they must obtain nourishment from a prepackaged form.
Yeah, all I had was a bowl of Cheerios. You look like a regular running deli here.
Yeah, but you don't need it. I need it. You're too light, yeah. It takes more energy for me to move myself around.
Nutrients are needed to build tissue like these finely honed muscles and to provide the energy needed to move them.
In the biology of life, what we do for energy is to take molecules and break them apart using the energy of the bonds that had been formed by various other chemical reactions, gather that energy and use that energy to create more molecules, to replicate and divide cells, to give heat, to give light, and so forth.
Respiration is the process by which the body harvests energy in an appropriate form.
The most important and exciting and efficient way of breaking down molecules and getting energy is what's known as aerobic respiration. That's where we take sugar, we take fats, we take proteins and amino acids, and literally burn them.
The burning of food molecules releases energy, some of which is trapped in the bonds of ATP molecules.
The currency of the energetic realm in life is ATP. No single creature has ever been isolated that lives that doesn't use ATP as the central energy exchange unit.
ATP is critical because when broken down, it releases just the right amount of energy to drive many cellular processes.
What we have to do is to suck out the energy from those bonds and conserve it as ATP. And we do that by breaking those bonds down, one step at a time. And as we break them down, we make these little steps in such a fashion that wherever it's possible, we conserve a unit of energy and build it into an ATP molecule.
During exertion, a person's need for fuel increases. One fuel frequently used by the cell is glucose, a simple 6-carbon sugar.
All right, feeling good so far.
Glycolysis is the process in which the sugar molecule initially breaks down and releases energy trapped as ATP and NADH.
If you want to start the breakdown of sugar in a yeast or human being, what you have to do is initially invest ATP molecules, 2 ATP molecules, to make the sugar into a phosphate ester, which in turn I can then break down by glycolysis to yield 4 ATPs for every 2 I invest. So it's a doubling of your energy money. It's a doubling of your ATP currency by investing the initial ATP. But you've got to invest that to begin.
The breakdown of glucose to pyruvic acid involves a series of reactions which occur in the cytoplasm of the cell. Most of the energy from glucose, however, remains locked in the bonds of pyruvic acid.
But there's an enormous amount of energy that's left in these molecules of pyruvic acid. So what do you do with those? And the answer is you put them into this wonderful machinery called the mitochondrion.
It's in the mitochondrion, the powerhouse of the cell, that the Krebs cycle breaks down pyruvate to carbon dioxide and water.
Aided by key enzymes, pyruvate, the product of glycolysis, passes from the cytoplasm into the mitochondrion. The pyruvate is stripped of energetic electrons and a carbon dioxide molecule and converted to a 2-carbon fragment. In the mitochondrion, the 2-carbon fragment combines with an existing 4-carbon molecule. A series of reactions rips carbon dioxide and electrons from the molecules and forms a 5-carbon, then a string of 4-carbon compounds. In addition to the release of carbon dioxide, the Krebs cycle generates an ATP molecule and transfers energetic electrons and hydrogen ions to the coenzymes FAD and NAD. These reduced, electron-rich coenzymes stand ready to provide the energy needed to produce the bulk of the ATP, harvested during aerobic respiration.
While 2 turns of the Krebs cycle net 2 more ATP, it transfers energy-rich electrons to the coenzymes NAD and FAD.
Best of all is that what happens as this oxidation takes place, we rip electrons, these subunits of atomic particles, we rip electrons off of these organic molecules and put them into this machine which is called oxidative phosphorylation. Electrons then are transported down this electrical gradient. So it's beautiful. You take hydrogen ions and you take oxygen and you take electrons at a high potential and you link them together, and out of this process comes oxidative phosphorylation, electron transport, and that makes roughly 90% or better of the energy that your body and my body uses to do all of the processes of life.
We're doing great. Looking good, feeling good.
Electron transport phosphorylation, the final stage of aerobic respiration, takes place in the inner membrane of the mitochondrion. This membrane is studded with enzymes and electron carriers, arranged in precise order. Electrons donated by the energized coenzymes NADH and FADH2, produced earlier in respiration, are passed along this transport system. Energy liberated by this process is used to create a gradient by accumulating hydrogen ions in the outer compartment of the mitochondrion. The passed electrons are eventually transferred to oxygen and joined by hydrogen ions to form water. Additionally, the energy of the hydrogen ion gradient drives ATP synthesis, a process which accounts for most of the ATP produced during the aerobic respiration of glucose.
In eukaryotic cells containing mitochondria, the complete breakdown of glucose produces 36 to 38 ATP, with all but 2 produced in the mitochondrion. With this ability to produce energy, mitochondria are similar to furnaces.
When I think of a furnace, I think of a machine in which I pour some sort of a fuel, whether it's oil or gasoline or whether it's coal, and I heat it up, it bursts into flame. And that flame is the process of ripping apart the molecules of the fuel and taking that energy and liberating it as heat. I think of a mitochondria in the same way, except I don't have to go to Fahrenheit 451. I'm at Fahrenheit 97 degrees, which is the normal body temperature. And now I'm still ripping the molecules apart, but I'm ripping them apart in little tiny increments, in little bits and pieces of bonds breaking, not explosively, but under complete control of the chemical reactions of the cell.
And just like a fire in a furnace, aerobic respiration requires oxygen.
Oxygen is necessary for all aerobic life. That is, life requires oxygen in order to be able to burn the fuels. The only creatures that can live without oxygen are the so-called anaerobic creatures, particularly yeast and bacteria. They cannot ski, they cannot surf, they cannot make movies, they cannot understand the nature of the universe. They don't have enough energy to do it. But creatures who have the ability to burn fuels in the presence of oxygen can gain enormous amounts of energy out of the foodstuffs that they consume.
4:11:36 is my time, and I ran the whole way, except for the water stop.
Brewmaster Tom Burns makes beer for a living, and like a baker or a winemaker, he relies on single-celled fungi called yeast to make his product taste good. He also relies on a process called anaerobic fermentation.
During the anaerobic fermentation, this is where ethanol is produced. These byproducts allow yeast to have all kinds of unique flavors that make beer unique in itself.
When one thinks of yeast, or at least when I think of yeast, I think of some creature lying in a bottle and making good red wine or some nice, wonderful, rich beer. In point of fact, yeast can live in aerobic environments, in the presence of oxygen. When a yeast lives in air, then the yeast lives just as we, human beings, live. They have glycolysis, and they have the Krebs cycle, and they have oxidative respiration, and they make ATP just like we do. But you can put that yeast into an anaerobic situation, and it will become a fermenting organism, making carbon dioxide and alcohol. You put us in an anaerobic environment, and you got a piece of dead meat.
Yeast can survive and even thrive in an anaerobic environment because they can continue to carry out glycolysis even when oxygen is not present to accept electrons. Instead, certain organic molecules perform this function with the production of alcohol and carbon dioxide. This is not an option for complex organisms such as humans because so little ATP is produced.
One is struck by the fact that if one had to live solely in the world without oxygen, you would have a very, very simple set of living systems like we see in algae and in some yeast and in some bacteria. They can't make very much energy from the chemical fuels that they burn. By a process of glycolysis, anaerobically, one gets only 2 molecules of ATP for every sugar that one burns. But if you think about aerobic respiration, for that same sugar molecule, now doing all the respiratory and electron transport mechanisms and so on, we now get 38 molecules of ATP per molecule of glucose. That's an enormous difference. That's 19 times more energy. It's no wonder that creatures have been able to evolve only in the presence of oxygen, because otherwise there just isn't enough energy to do all of the wondrous things that these complicated creatures that live on this earth can do.
Under anaerobic conditions, yeast produce carbon dioxide and ethanol, the alcohol that many enjoy, as byproducts. Interestingly, it's the escaping carbon dioxide that's responsible for the head on the beer.
The foamy head comes from the nucleation sites in the glass. And what happens is the nucleation sites, which are small imperfections in the glass, and what happens is the CO2 finds a place to actually pull out of solution. And once it grabs enough CO2, it grows a large enough bubble, and then the bubble creates a large enough that the surface tension can't hold onto the bubble, and the bubble rises to the top. So basically it means is if you have a dirty glass, it'll be very foamy, and if you have a very clean glass, you won't have so much foam.
Over the years, humans have used the ability of a number of organisms to anaerobically respire. Yeast are used not just to produce beer and wine, but to leaven bread. Other organisms anaerobically respire with the production of lactic acid, like that in yogurt and buttermilk, sauerkraut and soy sauce. Even human muscles produce lactic acid under anaerobic conditions for a brief period of time.
When that individual has been sprinting very, very hard and the muscle has used up all of the oxygen that's available to it, and as a consequence begins to produce lactic acid. You cramp up, is what it's called. And at that moment, when you stop, then oxygen flows in through the red cells in the blood. The lactic acid is then allowed to come back and be burned through the Krebs cycle. We cannot go for longer than 3 or 4 minutes without oxygen in our body, or we die.
All right, that's pretty tough.
It's interesting to think about the balance of life on Earth in the sense that plants gather sunlight, take carbon dioxide and water, and convert that to sugar by a process called photosynthesis. In the process of doing that, the plants have a mechanism to take that light energy and to convert it to ATP. In turn, the animals and other creatures on the earth take those pre-formed carbon molecules, the sugars, the fats, the amino acids, and so on, and they take oxygen, which the plants have made, right? And they literally burn those fuels, and that is fundamentally how we run our bodies.
Regardless of where it comes from, energy must be converted into a form that's able to drive metabolic functions. Through the wonder of photosynthesis, plants harvest sunlight, then turn that energy into fuel that's consumed by humans and other animals. In this way, life on our planet is an intricate, ongoing relationship of energy in and energy out.